So for active transport, if you cast your mind back to what we did in diffusion, all right, you remember that substances in diffusion are always going to move from an area of high concentration, all right, to an area of low concentration, okay? And it may or may not be through a particular membrane or barrier, okay? So under normal circumstances, it would go in that direction. The solute would flow from left to right in this case, all right? Now, remember we represented that as going, if you have two concentration bars, if you like, presenting it like that with, for example, that, and it's going down the concentration gradient, like that.
Yeah.
All right? That happens naturally, okay? So that down the concentration, gradient, right, does not need any help. And compare this like water flowing downhill. Does that make sense?
Yeah.
It will just do it because of the gradient, right? Literally just flow downhill. If, on the other hand, we want to make it go against the concentration gradient, and I'll give you an example of that as to why we might want to do that, all right? So if you have, for example, a cell, right? There's your nucleus, all right? And the cell already has lots of glucose in it, because what do cells need glucose for?
Energy.
Yep. And what do they do with it?
Respiration.
Excellent. Yes. All right. So there's your cell, and it obviously wants to get as much glucose as it can. So it already has probably a lot of glucose in its cytoplasm. All right. And then what you have next to it is, let's say, something like this. I mean, it's not the best diagram in the world, but it'll do. Okay. So you're going to have the blood flowing through here, etc. All right. So that's a capillary. And in that capillary, the plasma, the fluids, are going to contain glucose molecules, like that. All right? So what's happening here is that the cell wants more glucose. It always wants to take in as much glucose as possible, but the concentration gradient is going the wrong way, right? Under normal circumstances, the glucose would actually leave the cell and go into the bloodstream, which we don't want, all right? So two things prevent it. Right, so did that bit make sense, yeah?
Yeah.
All right, the important things to note here, the cell membrane controls movement of substances. Okay, so in this case, it stops the glucose leaving the cell, and instead what happens is energy is used, right? Because we are trying to, this time round, we want the, and I'll just do a small thingy diagram. This time we want to move it that way, right? We want to go from low to high, which is obviously against the concentration gradient, all right? And so need to move substances against gradient. All right. So exactly the same way that you would have like moving water, consider move water up a slope. All right. So if you want to move water up a slope or lift it up in height, what do you have to do? It's not going to do it naturally, is it? It won't do it on its own. So what do you need to do to it? The way to think about it is you're going to have to use energy and do work. Does that make sense?
Yeah.
All right. That is active transport. All right. So active transport is basically using energy to move a substance from an area of low concentration to an area of higher concentration using energy. Okay? So let me put that in. Moving a substance area of low concentration to area of high concentration using energy. Okay? And that's obviously against the gradient, the concentration gradient, right? Now, it's obvious that the amount of energy you're going to use to move the substance in, all right, obviously has to be less than the amount of energy you get from it. So when we're moving the glucose into the cell, we're going to use some of it in respiration, right, to release the energy that helps us move that substance in, all right? But the overall benefit, you still end up with more glucose that you can use for respiration for other processes. Okay?
Okay. Yeah.
Does that entire thing make sense so far?
Yeah.
All right. Any other questions on that?
No.
All right. There are more complicated bits to it in terms of how it gets through the cell membrane and so on and so forth. All right. But that's something you don't need to know just yet at this point. All right. The other bit that you wanted to know was surface area, right?
Yeah.
Okay. Let me explain that one. Now, this one's going to be a little bit tricky. Let me see if I can do this properly. If you take a simple cube, right, a cuboid, if you like, a 1 centimeter cube or something like that, and how am I going to draw this? I've got to think about how to draw this properly. Something simple like that, all right? Now, if that's a 1 centimeter cube, all right, then what is the surface area, the surface of each of the faces?
1.
Right, so what's the total surface area?
6.
Good. Total surface area equals 6 square centimeters, all right? And what's the volume?
1.
Right. There, all right? Cubic centimeters. Now, if I double that, okay, so I have something that sort of size. All right. Like that, right?
Yeah.
So it's all 2 by 2 by 2, that sort of thing. I know it looks a little lopsided, but it'll do, all right? Now, what's the total area of this face here, each of the faces?
4.
4. And you got 6 of them, right?
Yeah.
So again, the total surface area, 4 times 24, exactly, well done. And what's the new volume?
8.
Yes. Okay. Now, do you see the surface area to volume ratio?
Yeah.
For the first one is just, oh, actually, let me redo that. Surface area to volume ratio, 6 to 1. And over here, so that's just 6. And over here, you've got 24. Do you see how the ratio has decreased? Yeah?
Yeah.
So as the shape gets bigger and bigger, or the object gets bigger and bigger, the surface area to volume ratio, in other words, how much, I'm going to put it into biological terms, how much skin it has compared to how big it is, right? The proportion gets smaller and smaller. Okay? Right. One of the reasons for that is, look at this, and I will put this into a different context if you like, see if I can make this clear to you. When you have a small shape like that, the center is not very far. Do you see? The center of the blob is not very far from the surface. So there's like easier to get to the surface. If I now take a shape that's much bigger, like that, and I'm at the center, look at how much further I need to go to get to the surface. Yeah?
Yeah.
There is a lot more stuff in there that doesn't have access to the surface, if you want to put it that way. All right. So if you were to consider this for the purposes of, for example, heat loss, gas exchange, right? All of these things, or exchange of any substances, becomes more difficult in larger objects. Does that now make sense?
Yeah.
Yeah. So if this was, let's say, if I come back to my original diagram, so if you have a big space, like your lung, for example, if it were just one great big bag like that, all right, it would be less efficient. All right. Whereas if you have small pieces, then there's much more surface area for the same amount. So in other words, if I were to cut this, right, this whole volume of 8, and cut it up into 8 little cubes, okay, I would have a lot more surface area for the same volume. Does that make sense?
Yeah.
Yeah. So let's just demonstrate that. If we go from that some form of a cuboid, I know it's a bit rough and ready, but bear with me. I want to do this for you reasonably quickly. So that's your 2 by 2 by 2. All right. So volume is 8. Surface area, we've already demonstrated, is 24. Okay. And if we then cut it up into 8 times 1 cubic centimeter cubes, all right, then now we have, what's the surface area of each of those cubes that we established earlier?
1,
No, surface area of the whole cube.
6.
6, right? So you'd have 8 times 1 cubic centimeter cubes from the same big lump, but then you'd have 8 times 6 square centimeter areas, right?
Yeah.
So therefore, volume is still 8, but the total surface area is now 8 times 6, 48. All right? And so you're going back to a much higher amount of surface area for each bit of volume. Okay? So breaking up, and this is the crux of the whole issue, breaking up a large, what do you want to call it, shape, into smaller shapes, or just sizes, increases the surface area for the same volume. All right? And that, therefore, speeds up exchange. So call that diffusion if you like. Heat transfer, etc. Does that answer your question?
Yeah.
Yeah? Anything else you'd like me to explain on this? No? All right. One other benefit of this is, in chemistry as well. All right. If you want to react maybe a solid with some acid, like, I don't know, calcium carbonate or something with some acid, right? If you have a great big lump of it like that, then it's going to take a long time because the acid can't really get to all the solid. But if you crush it up into small pieces, little chips, or a powder, then the reaction gets so much faster because there is a lot more surface area for the reactants to come together. All right? So the same principle of surface area getting increased somehow has applications both in biology and also in chemistry. All right?
Yeah.
Any other questions?
No.
No. All right, then we'll pause there then.